our next speaker will be Heather Kudyba. And Heather, you can go ahead and share your screen. So just a little bit about Heather. So Heather received her undergraduate degree in biology before going to the University of Georgia for her PhD. So her work focused on using conditional tools in P. falciparum to identify and study the biology of essential genes during the blood stage. She then started her postdoc in UL Vega Rodriguez's lab in 2019. And here her interests are establishing a set of conditional tools to study essential genes in the mosquito midgut stages of P. falciparum. The floor is yours, Heather.
Thank you. Hi, as she said, I'm Heather Kudyba, and I would like to thank the organizers for giving me a chance to talk to you today about my work. And let's go to the next slide. So due to the haploid nature of Plasmodium falciparum, conditional knockout and knockdown systems are the best way to study essential genes in the parasite. And conditional systems exist in P. falciparum at all points of the central dogma, allowing for disruption in the parasite genome by conditional knockout at the DNA level, disruption and translation at the mRNA level, and direct inhibition of protein function based on the addition or removal of small drug molecules. The conditional knockdown and knockout tools, which are currently available to study essential genes and to interrogate the biology of Plasmodium falciparum, have primarily been utilized in the blood stages. However, few tools have been used in the mosquito midgut stages of the parasite. And these stages are prime targets for malaria interventions, as the mosquito dramatically reduces the parasite numbers by imposing the most severe bottleneck during the parasite's life cycle, as the parasite is exposed to the mosquito immune system and human serum components. Thus, for my postdoctoral work, I sought to employ the use of many of these conditional systems to study essential genes during the midgut stages of Plasmodium in the mosquito vector. So my work thus far, and what I'll be talking about today, has been focused on adapting the Ribozyme system to the mosquito midgut stages, which allows for knockdown at the mRNA level. And the way that this system works is that you insert the Ribozyme sequence after the coding region of your gene of interest. And following transcription, the resultant chimeric mRNA contains the Ribozyme. And this ribozyme is inactive in the absence of glucosamine. However, if glucosamine is added, it binds and activates the ribozyme, resulting in mRNA cleavage and therefore knockdown of protein expression. And this system was an attractive system for me to start with for many reasons. One, it requires minimal genetic manipulation to utilize the GLIMS system compared to other available systems. It's reversible, so once glucosamine is removed, protein expression can be restored. And there is a GLIMS ribozyme with inactivating mutations that was published termed M9 that does not respond to glucosamine treatment. And this M9 inactive ribozyme serves as a great control, especially with us trying to adapt the system to a whole other life cycle stage. So as I mentioned, the GLIMS system requires the addition of glucosamine to initiate knockdown. And one thing that we had to think about was the timing and delivery of glucosamine. And as you can imagine, there are many, many different permutations of when and where you could add glucosamine. The primary strategies that we have been utilizing for knockdown are to add glucosamine to mature gametocytes prior to feeding, or by adding glucosamine to the blood feeder, with the readout for mosquito midgut survival being oocyst development in the midgut. So the first thing that we had to determine was what concentrations can be given to the mosquito of glucosamine in the blood meal without being toxic. So to determine if feeding the mosquito glucosamine in a blood meal would have an impact on mosquito survival, we added glucosamine at concentrations of 0, 2.5, 7.5, 15, and up to 30 millimolar. And we then monitored mosquito survival for 16 days. And our data indicates that concentrations up to 30 millimolar did not have a substantial effect on mosquito growth. And the concentrations that are typically used in parasite culture do not exceed 5 millimolar, so these higher concentrations would be way in excess. So while we were trying to develop this system, we decided it would be useful to have a reporter line so that we could further validate the system. Thus, I generated plasmids where we have GFP tagged with the GLIMS ribozyme, or M9, which is the inactive ribozyme, and transfected into NF54. And once we had the reporter lines, we had to first characterize it in the asexual stages to make sure that the knockdown system is working as expected. So the first panel I'm showing is just IFA of these transgenic parasites that I created, showing that they are indeed expressing GFP. And we also wanted to make sure that when we add glucosamine to the asexual parasites, that we are getting a reduction in GFP protein levels. So to test this, we incubate the GFP, GLMS, and M9 parasites with glucosamine. We take and sapin and lyse parasites over a 72-hour period every 24 hours, and then run the lysates on a blot and probe for GFP, which is here, and EF1 alpha as our loading control. And in the GLMS line, we do get this nice degradation in GFP protein levels as expected, but we do not see this in the M9 control, which is great. I also took live images of these parasites during gametocytogenesis in the presence of both 2.5 and 5 millimolar glucosamine. And these are day 9 gametocytes. And as you can see that when we're incubating with glucosamine in the GLMS line, we do have a reduction in GFP expression, but not in the M9 control. And while the images that I'm showing here, it's not quantitative, I thought it provided a nice visual representation that we are getting knocked down in these stages. So the experiment that I'm going to show here is condensed from almost a year of experiments where using these lines, we have determined what concentrations are tolerated by the parasite in the midgut when given in the culture, when given in the blood feeder itself, or the combination of the two. And we have these 3 conditions that I'll walk you through in the graph. So we have here the first, let's not skip 100 slides, okay. Okay, we have here the first column is the control. We then have glucosamine added to the gametocytes for 48 hours prior to feeding to the mosquito at 2.5 millimolar. And then we also have where we have glucosamine given in the blood feeder at the time of feeding to the mosquito at 0.5 millimolar. And then the last panel is the combination of the two. And then 8 days later, we dissect and count leucis. So what I'm showing here is that just in summary, is that the culture and feeder concentrations individually are well tolerated by the parasite. However, when these conditions are given in combination as toxic. And this is strange as glucosamine is removed from the mature gametocyte cultures prior to feed. But because you have this M9 control line, if you were to have high enough infection levels, you may be able to use the combination of glucosamine in the culture and feeder and compare it to your controls that way. And our experiments now are including doing QR-to-PCR to demonstrate that we are knocking down GFP in the midgut upon glucosamine addition. So while we were working on adapting the GLMS system to the mosquito, my old lab mate from my PhD lab, Manuel Fierro, published a paper on an ER resident protein and its role in the asexual development of P. falciparum. And he used the GLMS system to demonstrate that this ER resident protein was essential for asexual growth. So this is just a figure from his paper. He has the GLMS M9 lines. The M9 grows fine in the presence of glucosamine and the GLMS does not. And so this is published work. So just briefly, his work showed that in Plasmodium falciparum, this protein was required for egress and invasion. And he found that the death phenotype that he observed in the asexual stages was due to an inhibition in proteolytic processing of several published proteins shown to be required for egress and invasion. So this protein became interesting for me to study in the midgut stages for many reasons. Not only did he use the GLMS system, which is what I'm trying to adapt to the mosquito, which I'm trying to adapt to the mosquito, PIERC, and all of the proteins downstream whose proteolytic processing was affected during ERK knockdown are highly expressed in the ukini based on data available on PlasmoDB. And additionally, studies have indicated that plasmepsin 10 and AMA1 that are in this pathway are required for establishing an infection in the midgut of the parasite. So hopefully I've convinced you that this is a very interesting protein. So the first thing that I had to do is remake the ERK, GLMS, and M9 cell lines using CRISPR-Cas9 gene editing in NF54, since Manuel's lines were made in 3D7, which do not form infectious gametocytes. So when tagging the GLMS, with the GLMS, we also appended an HA tag to the 3' end of the gene so that we can monitor protein expression, subcellular localization, and perform co-immunoprecipitation assays. And we were able to obtain 2 independent clones for the ERK GLMS line and 1 for the M9, and this was confirmed by a PCR integration shown on the right. Using the HA tag, I performed colocalization assays with ER marker BIP in all of the lines. These are the asexual parasites, to show that just like in Manuel's paper, that this protein is localizing to the ER. And on the right is the growth curve of the asexual stages in the presence of glucosamine. The M9 is in orange and the GLMS is in blue and green. That the M9 is unaffected by glucosamine, but the GLMS parasites fail to thrive in the presence of glucosamine. We also wanted to determine if in the asexual stages that we are achieving knockdown by performing Western blot analysis with the ERK, GLMS, and M9 lines in the presence of glucosamine. So as before, we incubated them over a 72-hour period in the presence of glucosamine as before. And we did indeed see a decrease in ERK protein levels, which is in red, but not the M9, which is in blue. So that was comforting. And we next wanted to see if ERK was required for gametocytogenesis. And this is more tricky because ERK is required for asexual development. So we cannot just remove glucosamine at the start of gametocyte induction. So for this experiment, we induce gametocytogenesis and then add glucosamine at day 7 or day 10 of gametocytogenesis, and then calculate gametocytemia at day 16 when the gametocytes are mature. So on the left is the GLMS and the right is the M9, and we did not observe any significant difference in gametocytemia between controls and glucosamine-treated parasites. And then we wanted to see if adding glucosamine, if this protein was required for establishing an infection in the midgut. So the first thing that I'm showing here that we've tried is feeding mature gametocytes to the mosquito in a blood feeder containing glucosamine at 0.5 millimolar. And then 8 to 9 days, we looked at oocyst numbers. And remarkably, we observed a significant decrease in oocyst numbers, about an 83.3% inhibition produced in the ERK GLMS parasites when glucosamine is added to the feeder. And this difference was not observed in the ERK M9 lines, which suggests that ERK is required for establishing an infection in the mosquito midgut. And this is the first time that this system has been used in the midgut stages of P. falciparum to our knowledge, and we have used it to identify an essential gene required for a midgut infection. So as you can imagine, we have many future directions and ongoing experiments. So the main thing is assessing the level of knockdown that we achieve in the midgut for both the GFP GLMS M9 lines as well as the ERK GLMS M9 lines. I'm working to do a series of experiments just looking at subcellular localization of ERK throughout all of these different developmental stages of Plasmodium falciparum. We're looking to quantify the eukinates in the ERK, GLMS, and M9 lines during knockdown. We're also working in collaboration with the Leiden Malaria Research Group to generate Plasmodium berghei promoter swap lines so that we can really better study the biology of ERK in the mouse model. And there are several experiments we can do with that, including eukinate and oocyst development, as well as Western blots to look at their defects in processing of downstream targets, as Manuel saw in the asexual stages. And the last thing is that we've been working on is to determine if ERK is required for invasion and or egress from the liver cells. And so this is just my last slide. And basically, as I said, that we are interested in whether ERK may be required for invasion or egress from liver cells. And studies using an AMA1 inhibitor have suggested that AMA1 is required for invasion of the liver. And likewise, studies using plasmepsin 10 inhibitors have demonstrated that plasmepsin 10 is required for egress from the liver cell, which are 2 of these proteins that are downstream of ERK. So in collaboration with my husband, who works at Alison Roth's lab at Walter Reed, we are testing our mutant ERK lines on their ability to invade and egress primary human hepatocytes. And so far, just what I'm showing here is that we have ran a hepatocyte toxicity assay to glucosamine, and even at 10 millimolar, the hepatocytes have not been affected. So with that, I would like to thank my lab. I think every single person in the lab has contributed to this project at some point. I would like to thank my PhD lab, Vasant, Manuel, and Carrie. They're a great support. I talk to them all the time about this project. Allison Roth and my husband Carl at Walter Reed, the Leiden Malaria Research Group, the insectary staff at the NIH and at Johns Hopkins, and the Marcelo Jacobs Loreno lab for their help. And I'll be happy to take any questions.
Great, thank you so much, Heather. While the questions come in, I wanted to see if you could maybe describe a little bit more about the reversibility of the glucosamine. So I guess how long would it take to like leave a mosquito system or something like that?
So that's a little bit hard to determine. I mean, we haven't determined it yet, but I think the kinetics would be a little bit slow. Like the knockdown will be a little bit slow because you have all the protein around and you're knocking down to the mRNA level. But once you remove glucosamine, I think it's, I think my PhD lab, they've observed it in about 4 hours in asexual culture. So yeah, it's just something that we haven't looked into yet, but.
Great. And then another question, you mentioned that if you give the glucosamine in combination, it's toxic. Could you just lower the concentrations in the in culture and then in the feeder?
Certainly, yeah. So that's also some experiments that I've done. Adding all these different permutations of glucosamine at different stages. Yeah, I think that you can lower it in the culture or the feeder, and that might kind of balance it out a little bit.